Good evening. I'd like to welcome everybody to our spring series of Boston Seminar Series. For those who don't know, we do this every 4 to 6 weeks or so with MIT faculty and alums presenting their work to the community. And I'd like to just, before we move on to Paula's talk, just want to invite you to our next event, which we'll be hearing from Cognitive Behavioral Sciences Professor Earl Miller on April 18th at the Harvard Club of Boston. His talk is titled Working Memory 2.0. He'll teach us all about executive control, function, and dysfunction. After that, on May 1st, Marty Culpepper and the CEO of the MIT Alumni Association, Whitney Espick, will share some of their work with us, also at the Harvard Club. And if you want to get involved with the Club of Boston, we are always looking for more people to come and help out and join the fun. Our annual meeting will be May 16th at the Broad Institute, and the speaker will be Hyundai Park, president of the MIT Alumni Association, MIT alumna herself. And again, that'll be May 16th at the Broad. And thank you all for coming again. I'd like to hand over the mic to Bonnie Kellerman, who will introduce our speaker.
Well, welcome everybody. So it's my distinct pleasure to introduce tonight's speaker, Paula Hammond, who is a faculty member as well as an alum. I've actually known Paula since her undergraduate days at MIT, so it's been great to see her trajectory. She graduated from MIT class of 1984. She went on to get her PhD from MIT in 1983, both in chemical engineering. She spent a few years up the road at places we won't mention doing her postdoc, but then had the good sense to come back here at MIT. She's now the David H. Koch Professor of Engineering and also head of the Department of Chemical Engineering. She's a member of the Koch Institute for Integrative Cancer Research. She's also a member of the MIT Energy Initiative and a founding member of the MIT Institute for Soldier Nanotechnology. Professor Hammond's been elected to the National Academy of Engineering, to the National Academy of Medicine, to the American Academy of Arts and Sciences. She's received numerous awards and prizes, but if I spent time telling you about all of the awards and prizes she's received, we wouldn't have time for tonight's presentation. So I won't tell you about her awards. I will, without further ado, invite Professor Hammond to come to the podium to share information with us about her research. Welcome.
Thank you so much. We're going to go ahead and get started here. No, it's fine. It's the button wasn't working. There we go. All right. First of all, thank you for the chance to speak to you today. This is a wonderful setting because there are people I've known here for many years, including going way back to when I was a student, and people who I'm meeting for the first time, and we all share that excitement that we got at MIT for science and technology. I'm going to be talking today about the work that we're doing in our lab that uses a very simple technique called layer by layer or electrostatic assembly. And this method is something that we can actually use to address a broad range of biomedical applications. Everything from biomedical implants and wound dressings down to cancer-targeting nanoparticles. So the way this process works is that you start with the substrate, and it can be virtually any substrate, glass, plastic, a range of different films. And that substrate, as long as it has some sort of initial charge on the surface, can be immersed into a dilute aqueous solution that contains a multivalent charged species, any sort of material that has multiple charges. That charged species is going to absorb onto the substrate until ultimately that surface charge is reversed. So if you had a negatively charged substrate, you immerse it into a dilute water solution that contains a positively charged material, you'll get something that is positively charged in the end. You can then rinse the substrate to remove anything that is not truly absorbed onto the surface and immerse it once again into a water solution that contains, once again, something that is oppositely charged. So you can alternate positively and negatively charged species back and forth in series and continue to do this ad infinitum. There's no theoretical limit to how many of these layers you can put down. Now, what you're absorbing is a monolayer that's fairly thin. It's only a couple of nanometers in size. So on a molecular level, we're thinking essentially of something that is in the range of 1/10,000th of a human hair. When you put down multiples of these layers, you can actually change the average thickness. So typically in our work, we're working with materials that build up at 100 to 200 nanometers per layer, which means we can get to microns fairly rapidly. When we're building these films, we can incorporate virtually anything into the film as long as we follow this rule of plus alternating with minus. That means that we can incorporate a very broad range of materials into the thin film, and I'll explain why we will want to do that in a minute. But I should point out that you can also use other kinds of interactions. For example, systems that undergo hydrogen bond interactions with each other can also be alternated in these films. So you can have a material that has a hydrogen bond donor alternated with a material that has a hydrogen bond acceptor. And we've used these kinds of approaches to incorporate a range of different biological systems as well. This is actually very interesting to us for drug delivery for several reasons. And I'll try to explain that in a little bit more detail. But the general idea is that we're working with a water-based solution, which means we can incorporate a range of different therapeutics that normally we couldn't incorporate into a thin film, including proteins, nucleic acids like DNA, siRNA, and a range of other biologic drugs that typically are difficult to encapsulate in a polymer without destroying their activity. Using water, we can actually keep the processing conditions very safe, very mild, and very easy to implement. Now, the big picture is that we can use this to coat a range of different biomedical implants and surfaces with drug-containing thin films that can undergo controlled release. Now, you might compare that with a more traditional approach. For example, polyesters. And polyesters is one of my favorite materials. I'm a polymer scientist. And my group sometimes call themselves the polymers, which is some sort of play on my name. The idea is that you can take a conventional coating that might be a polyester like polylactic glycolic acid. It will degrade in a very controlled fashion. But because it is a polymer and you're trying to create a blend with, let's say, a protein-based drug, it turns out that it's going to phase separate at very low concentrations of the protein. And the reason for this is that when you have a large molecule like a polymer, and you blend it with something else, that gain in entropy, going way back to thermodynamics, you remember that when you mix two things together, they want to mix because you increase entropy. But when you have a polymer chain, you already have these units linked together to create a very large molecule. So when it blends with something, you don't get as much entropy. So they don't want to mix as much as most materials do. So you end up only being able to put about 1 to 2 weight% of drug in before you phase separate. And if you phase separate, when you release the drug, it releases in a very rapid and short time frame. You don't get control. So you tend to use these materials to release small molecule drugs that are hydrophobic and will not be needed in large quantity. The other problem with a conventional coating is that if you put multiple things into that conventional coating, they all come out at the same time. So if you want to deliver 2 or 3 different drugs and you want them to have each their own very unique release profile in the body, you can't do that with our typical polyester or other kinds of degradable polymers. On the other hand, with layer by layer, we have several advantages. First of all, there's the whole thermodynamic idea. Thermodynamics went against us when we were trying to make a blend, but if we're using charge as the regulator, then all we need is to alternate charge compensation rules. That means that if I take a degradable polymer that's water soluble and charged, like these here, here's a collection of positively charged polymers. They are also polyesters, but they have not only a degradable unit, but this charged unit, they can dissolve in water and be layered with a different drug molecule. I can change the rate of degradation of the polymer by changing what I put into the backbone. So these are polymers that degrade at increasingly slower time frames. I can layer that with, let's say, a negatively charged biologic drug. You can imagine I can build up layers not only of just one drug, but of 2 or 3. I can even generate films in which they're stacked so that I have drugs 1, 2, and 3 in some sort of surface-based order. What this means is that, first of all, depending on which polymer I decided to pair with which drug, I will have a release profile defined by the degradation rate of that polymer. So now I can have more than one different release rate. On top of that, if I'm able to successfully create some degree of separation between the drugs when they're layered into these films, then I might be able to have a surface-based corrosion, and that would allow me not only to have them release at different rates or speeds, but also to have them release with a staggering in the time frame that they get introduced into the body. So the general idea here is that you might be able to have an intelligent thin-film coating that can release drugs at different times. Now, where would we want something this sophisticated? It turns out that there are several applications in which the delivery of multiple drugs is desired. And this is one of the reasons why this is actually a very exciting approach, because whether or not you want to control the relative rates of release, it's often very difficult to combine 2, 3, or more different drug types into a single polymer without creating a mess, because they really don't want to interact with each other. But because you're using charge to introduce these materials into the film, you can actually get a very nice controlled set of materials that release. Now, in this example, we're showing the release of a small molecule like a painkiller, another small molecule, an antibiotic, and a protein drug, or biologic, a growth factor in this case. And I'll describe the role that these growth factors might play. But you can imagine that from a biomedical implant, you might want to immediately address pain and infection. But over a longer time frame, introduce growth factors which might influence how tissue grows around the implant. So I'm going to give 3 examples of how we use this layer by layer technique, and I'm going to go from large to a little bit smaller to much smaller as I move along. I'm going to start by talking about bone regeneration. We've actually been using this method as a way to generate bone around an implant, or bone where bone cannot grow in a large defect, for example, a cranial defect. What we're interested in doing is releasing a natural protein, bone morphogenetic protein 2, or BMP2. This is a growth factor that actually induces the stem cells that reside in our bone marrow to arrive at the site and begin to differentiate into bone cells. It's an extremely potent growth factor. And in fact, it has the ability at very small amounts, nanograms to micrograms, to induce significant amounts of bone. It's actually a wonderful growth factor and has already been developed into an FDA-approved drug product called Infuse. Infuse is for spinal cord injury repair. It's a collagen sponge. You incorporate the BMP2 into this collagen sponge, and the surgeon places it at the base of the spine where repair is desired. But there's a problem with this product and with other products like it. BMP2 is not only very potent, but it also has the ability at large doses to stimulate nascent cancer cells and to grow bone where you don't want bone. It turns out that because it's incorporated into a collagen sponge that doesn't control release, they have to incorporate large amounts of BMP2 a few orders of magnitude more than you really need to have at the base of your spine. When it's incorporated and the surgeon sews up, most of the BMP tube flushes through the body within the next 8 to 12 hours. Most of it ends up in the bloodstream and finally excreted, but some of it hangs around and generates these undesired effects. You have enough left over at the base of the spine to regenerate bone, but you also introduce some risk. So for that reason, Infusion products like it have a black box warning, and it's only used in more extreme cases. It would be beautiful if we could incorporate just enough BMP2 to get bone regeneration, but only in the site of interest. And so the idea here is that if we can have a localized and very slow release of this drug at low doses, we may be able to do that. There are other growth factors that play a role in this bone regeneration process and in the regeneration of other tissues. For example, VEGF is a growth factor that induces the generation of vascular systems. And PDGF is a kind of partner growth factor that helps generate proliferation of these blood vessels and actually is a support, provides a support mechanism for vascularization. Now, these are proteins that have very real therapeutic value, but to us, in the end, they're just really complicated looking polymers. And we actually will look at a protein like this, understand what its isoelectric point is, the point at which it undergoes a switch from positive to negative, and then, based on that, we'll know whether or not we can treat it like a positively charged polymer or a negatively charged polymer when we introduce it into our layer-by-layer films. And these growth factors happen to be positively charged at pH 7, and so we treat them like a positively charged material in a layer-by-layer film. So here, these circles represent the protein. We can create a sandwich layer that is a tetralayer with a negatively charged polymer, or polyanion, the protein, another negatively charged polymer, and then one of those degradable polymers I just mentioned on the other slide. And that negatively charged polymer can be a number of different things. We've used everything from very simple polymers like polyacrylic acid, which is used as a food modifier and a viscosity modifier, and is generally regarded as safe. And we've also used natural molecules like the glycosylaminoglycans that reside in our body, a number of polysaccharides like dextran sulfate can work. So that's the general idea of how we package these proteins. Now let's talk about how we can apply them. In this example, we were interested in craniofacial repair, being able to use this kind of approach to regenerate bone where it's missing. And it's possible in a large number of critical injury and traumatic injury cases that large pieces or fragments of bone go missing. You can imagine a blast injury, for example, that can result in the loss of jaw, the loss of skull. There are also some birth defects which result in missing bone in the head or skull. We're interested in being able to grow bone in these critical defects that are so large that the body can't bridge the bone by itself. Right now, what's done, the standard approach is to introduce a template, which may be a plastic implant, or typically it may be titanium, and perhaps to introduce a mixture of ground bone graft that comes from the patient from another part. The favorite is to take it from the pelvic bone. And the cells that still reside in this area can be kind of plastered in, and you can try and regenerate bone in this way. This is an approach, but it is not necessarily the optimal standard of care. If we can eliminate the need to take bone from the patient, then we can decrease the amount of morbidity that takes place. Some of you, I know I have an example with my own mother-in-law who had a portion taken out of her hip to repair another part of her body, and that it took quite a long time for her to heal. Often we're dealing with patients that are old or may have their immune system compromised, so this is really not optimal. What we'd like to do is replace that kind of graft surgery with a much simpler approach by generating first a degradable membrane, and we'll use a familiar polymer like the polylactic glycolic acid polyester that I described before, and we'll process that polymer so that it's very porous, and we get these little foamy inserts. They're kind of a thin foam insert that degrades over time. We'll take that known polymer and then we'll coat it with our layer by layer film. So down at the bottom, you can see that that porous membrane now has a little top coat. This is a side view. You can see the top of the film here, and in the cross or cutaway, you can see that it's sort of sitting on top of this porous membrane. It's a very thin film, but we can pack a large amount of growth factor into a tiny amount of space. Because of those rules of alternating charge, rather than having 1 or 2 weight percent as we would in the PLGA itself, in our case we have somewhere between 30 and 40% weight loading of our protein in the layer by layer film. Now in this study, Nassar, the young man, I think we just flashed his picture a minute ago, and I forgot to mention this is the work of Nassar Shah, who finished up a couple of years ago and is now going to be a professor at UC San Diego. What Nassar was interested in was whether or not we could actually deliver 2 growth factors in a meaningful sequence. And the reason he was asking this question is because when you regenerate bone, typically with just BMP2, if it's a very thick amount of bone, it often doesn't have a very highly vascularized nature. You don't have blood vessels going through the bone. And because bone is, of course, a living tissue, this means that that bone can go necrotic, it can become embrittled. There can be other problems that are generated from having this bone not have a very highly vascular arrangement. On top of that, we thought it may be helpful if we generate the blood vessels then first and allow the bone to grow around those blood vessels, because if bone is generated first, it would be very difficult for blood vessels to find their way into that new bone tissue. So he decided to put PDGF, which is one of those growth factors I mentioned earlier that helps to accelerate and generate vasculature, as the top linkage, and BMP2, which is the bone generating system underneath. And he gets a release profile that looks like this. Here you can see the time in days, and here you can see the amount, cumulative amount that has come out of the film. And you can see that all of the PDGF is out in about a week. And we're getting a very nice, for us, low dosage of nanograms per milligram. You can see the BMP2 comes out in a period of somewhere between 3 and 4 weeks. And so it's much slower. And this is the kind of staggering that we like to have in this system. So when Nassar did this study, there are a couple of ways in which we can look at what's happening. We use a cranial defect model in which we generate a defect in the skull of a rat, and we use a control to show that this defect cannot close over time. We then use micro CT. So here you're looking essentially at a kind of X-ray of these defects over time to see the fill-in of bone in the matrix. So this is the first test. And we looked at 2 different doses of BMP2 by itself, 0.2 micrograms and 2 micrograms. We also looked at BMP2 at the low dosage with PDGF on top of it, again at a low dosage of 0.2 micrograms. And you can see that the bone fills in for all 3 cases, but perhaps just a little bit faster for the dual combination. If we, however, take a closer look, we can do histology. We can actually take sections of that bone and look at them after staining for blood vessels and bone in the microscope. And what we can see is that the untreated defect, of course, has this big gap in it. So these are the edges of the skull, and this is where bone does not exist. If we just put a membrane of PLGAN with nothing on top, you don't get bone, but you get this kind of bridge of collagenous tissue. It's kind of like a scab. Fibroblasts will crawl across the membrane, deposit collagen, but it's a very weak kind of matrix that it forms. So it's not any kind of load-bearing tissue. If you introduce just a little bit, the 0.2 micrograms of BMP2, you get a lot of bone. So this just goes toward the idea that dose, when we're working with very controlled and localized release, really is not an issue. We can stay down at these very low levels. But we see that the bone is not very well formed or shaped. It looks, you know, for lack of a better word, lumpy. And when we introduce 2 micrograms, we increase the dose by a factor of 10, we get a bigger lump on the head of the rat. And you can see that there are some fairly large defects and irregularities along the surface of the bone. However, if we introduce the BMP2 PDGF combination, where PDGF is on top, we see that we get bone that looks very much like the matrix itself on the edges, bone that looks like the native bone. We get a very nice vascularized system throughout the bone, and on top of that, we get this very nice smooth bone surface. And we believe that this is due to the fact that we have not only generated vasculature to provide nutrients to the bone, but that those blood vessels provide a pathway for a second set of bone cells that are called osteoclasts. And osteoclasts, unlike osteoblasts, are designed to deposit a protein or an enzyme that breaks bone down so that they can actually realign and perfect the bone. So they're the perfectors, and we get essentially what is called remodeling of the tissue. And that remodeling is what allows this tissue to essentially form this much nicer aligned arrangement. Now, what I'm not showing you are mechanical tests. We actually do mechanical tests on these bones. We put them in a mechanical tester pool, look at the tensile strength, we look at impact strength, and it turns out that in these first 2 cases, the bone is less perfect. It doesn't have the wonderful mechanical properties of native bone. But in this last example, we're able to match the mechanical properties of native bone in terms of impact strength and tensile strength. Finally, we look at how well oriented the bone is, and we find that in this last example, we get very highly oriented bone in comparison to the other 2 examples. So if we go back to that idea of being able to use the platform to deliver multiple growth factors, in the example of the cranial defect, we can release 2 growth factors that to us look very similar, but to nature are quite different. But we can also use the ability to introduce dual components to use a very small molecule, which is synthetic and man-made, and combine that with a growth factor. So in this case, the idea was that we might be able to generate an orthopedic implant coating. This is for whole joint replacements. Now, it turns out that the number of whole joint replacements, that's hip, knee, shoulder, some of you may have one or may have a good friend who has one. These have increased significantly over time for multiple reasons. One is that our technology for these implants has improved incredibly. So doctors are recommending this surgery more frequently and to a younger group of people. As we grow older with these implants and as we maintain our active lifestyles, which include anything from running and walking on a daily basis to mountain climbing and jumping out of planes, there's more of an opportunity for aseptic loosening of these joints. And what happens is that typically between 10 and 15 years, there needs to be a replacement. The first time they do the surgery, in the US and in countries like the US, there's typically not a concern about infection here. But after the implant has been present for about 10 to 15 years, when it's time to put in a new implant, there is some real concern because you have had this sort of area where infection can exist for extended periods of time. So when they do that replacement surgery, they'll remove the old implant, they'll essentially put in these antibiotic beads that release for a period of 6 weeks while the patient is braced, and then scrape out those beads in a second surgery and insert the new implant. So it's a 2-step, tedious process. It requires a huge amount of hospital time and extended care, and of course, a high cost. So what we're interested in doing is replacing that with a single shot, releasing enough antibiotic that we can eliminate an infection, not just prevent one, which means that we need significantly more antibiotic than what is used in the common sort of bone cements that are used by orthopedic surgeons. And we're also interested in releasing growth factors that will allow regeneration of bone. And here we'll focus on BMP2. So in this project, Juha built on the Sark's work with the incorporation of BMP2. And her goal was to create a dual release system in which she had BMP2 represented by the green color in this slide here at the bottom. And on top of that, introduced gentamicin sulfate, which is a positively charged small molecule that's a commonly used antibiotic. So the antibiotic is on top and the BMP2 growth factor is on the bottom. She decided that she wanted to separate these two sets of layers with a barrier layer to prevent mixing of the two molecules during the construction of the film. The more you're able to do that, then the more separated the rates of release can be when you generate your final film. So she used this nanoclay. It's actually a silicate, just common silica, but it's in this clay form in which you have a disc shape, just a nanometer in thickness, anywhere from 25 to a few hundred nanometers in diameter. And these negatively charged clay discs will absorb very nicely from water as we build up a film. It turns out that there are a whole range of lapinite clays that are out there, as well as moncorolonite and other clays. A number of them are regarded as safe by the FDA and have good biocompatibility and interact well with cells and even enable cell adhesion. So I don't know if you can see this as well, but on the left, what you're seeing is the growth, what we call the growth curve, when we're measuring the thickness of the film as we put down more and more layers of BMP2. And then we see the thickness of the film as we introduce some really thin layers of this lapinite clay, just a small number of them. Finally, we build up the gentamicin sulfate, and if we want, we can even put a second set of lapinite layers on top of the gentamicin sulfate. And now we have this very nice separated arrangement. Now, with this level of light, you may not be able to see it as well, but underneath what we're looking at is electron microscopy cross-section of Juha's film. And at the top you can see the silica as bright. In the middle you can see the lapinite again as bright. And then at the bottom we used a glass substrate, so it's also bright. And the intermediate darkness, these are the drug-containing layers. If we use EDS, we can actually label the silicon atoms. And so we have red, black, and then red. So what we're really showing is that we've been able to create this very well-separated set of drug-containing layers. When Juha looks at the relative rates of release of these two systems, what she saw was essentially that without any kind of capping arrangement, the gentamicin, which is always indicated by red, over a period of days, this is out to 70 days here, is released in a very short time frame. And the BMP2 is a little bit slower, but it comes out rather fast. So when we look at just the first 5 days, a good amount of both drugs are kind of out there, although there is some separation between the two. To increase the separation, if we just introduce the intermediate lapinite layer, we find that the gentamicin sulfate is still rapid in the rate that it comes out, but BMP2 is delayed significantly. So in that first few days, there's much less of the entire amount of BMP2 that's come out, and we actually get about a 14 times longer release half-life. Finally, with the antibiotic, we really like to be able to have enough present in the beginning to eliminate infection, and then a small amount that continues to be present over a period of 1 or 2 months, so that we maintain the antibiotic above the critical concentration needed to kill any new infection that might show up. So to do that, the capping layer actually allows us to accomplish still a rapid release, but slower, enough to eliminate infection, but you get this very nice extended stream out over a period of 60 days for the antibiotic to continue to release. So this is one of our ideal arrangements. And this is an example of a model in which we tested this system with an implant. So to explain these images, essentially this is again a rat femur, and we have the shape of the cross-section here, and there's a hole or defect that's drilled in, and then an infection is established within that drilled defect. So we actually have osteomyelitis, for the technical name, this bone infection, which is very highly established. It creates a biofilm. After waiting enough time for that infection to be established, we will introduce an implant. In some cases, we look at untreated implants, and we look for the regeneration of bone around that implant. Instead of bone regeneration, we see that although some bone tries to get deposited over time, we're really seeing a lot of resorption of bone. Here you can look down at the micro-CT and you can see where the bone is getting kind of etched away because of the byproducts of this infection. If we just have gentamicin present, but no growth factor, we're able to fully eliminate the infection, which is a good thing. And by eliminating the infection, over a long period of time, bone is slowly trying to reestablish itself. And you can see that here with the G representing gentamicin. However, we're not quite able to completely fill the cavity with bone. There's still some residual damage that has been done by the presence of that infected tissue. And it's going to take a while before we regenerate bone in this area. However, if we have the combination of BMP2 and gentamicin sulfate, we can completely fill in the bone over a period of 5 to 8 weeks. So we greatly increase the rate of recovery of patients in this way. So this is an area that we continue to be excited about. Now we're looking at whether or not we can deliver DNA that will encode for BMP2 as opposed to the BMP2 protein itself. And we're going to compare the results of those two to determine which is the more effective treatment. There are reasons why DNA is actually cheaper, easier, and is actually cleaner in terms of being able to generate a system that doesn't introduce something immunogenic. Okay, so I'm going to move on to the second example. This will be a shorter one. Really go from hard tissue to soft tissue because we think that we can use the same kinds of approaches to deliver biologic drugs to wounds. And for a typical wound, this may not be necessary. However, there are wounds that exist that have some measure or degree of dysregulation and therefore are difficult to close or undergo some other kind of dysregulated behavior. The best example of this are chronic wounds. In chronic wound healing, there are overexpression of some genes due to some sort of shift or change in the metabolism of the patient. Now this may be, and often is, due to hyperglycemia. So diabetic patients will have or experience diabetic foot ulcers, and this tendency to not close the wound is often due to a change in the expression of a range of different growth factors. It's actually a very complex process. That said, it's been thought that if we can correct the overexpression of even one or two of these genes, we may be able to enable regeneration of tissue. Sanofi actually came to us to talk about this problem. They are very interested at the time in being able to generate a wound dressing that would enable closure of diabetic wounds over relatively short time periods. In fact, they defined a 2-week period in which they would do this work. And one of the things we talked about was what we might deliver to address this. We decided that although it's interesting to look at proteins, and in our work we did look at growth factors that can accommodate and help facilitate wound closure, it would be very interesting if we could deliver silencing RNA or siRNA to turn off the genes that are overregulated in these conditions. So siRNA essentially is a nucleic acid that guides the body to essentially degrade or break down the ability of the expression of that gene. So if we have siRNA against a given target, we're going to silence that gene. And one that we were interested in was MMP9. This is actually a collagenase. It breaks down collagen. It has a role in remodeling tissue, and we talked about how important that was in the bone example. However, MMP9 should show up in larger concentrations later in the wound healing process. Early in the wound healing process, you're going to want to build up collagen. You need that extra tissue, often called granulation tissue, to close the wound. It turns out that in diabetic patients, MMP9 concentrations are much higher than they are in patients who wound normally. So the question is, can we deliver siRNA against MMP9 from a bandage? Now, this sounds simple enough, but siRNA is very susceptible to degradation itself. It's actually a very sensitive molecule. There are a number of RNases that are present in our bloodstream. They're protective. They're meant to address any sort of foreign body that is trying to evade us. But because RNases are present, it makes it difficult for us to deliver siRNA directly to a wound. On top of that, wound cavities are more aggressive than other parts of our body. The fact that they're undergoing the inflammation process means that there are a number of different compounds, chemicals that are present that are also designed to break things down. And they're also in a more acidic environment. So if you think of a wound environment, there's a lot going on there, right? And part of that is a slight lowering of the pH in that area. All of those factors make it more difficult to keep any kind of active component alive. So the idea here is, can we package siRNA into a bandage in a way such that it's able to be delivered directly to a wound without being deactivated? So this is the way we did this, and this is work done by my student Stephen Castleberry. Stephen is now working in industry in Chicago. Now in this work, he's generating layer-by-layer films, but he's actually doing two different kinds of films. He found that a very simple polysaccharide known as chitosan, which is derived from crab shells and has these positively charged groups, these amine groups on it, they actually work very well in layering siRNA, which is strongly negatively charged. So he can make siRNA and chitosan multi-layers, and these layers will be very stable. They're very effective at protecting siRNA. The only problem is that chitosan doesn't break down to anywhere near the time frames that are relevant to this wound healing project. If you're interested in breakdown over a period of a few weeks, chitosan will remain stable in the body for a few months. So underneath these layers of siRNA and chitosan, Stephen built a second set of layers that were designed to break down. And these contain one of the degradable polyesters we talked about. This one's called poly2, layered with dextran sulfate, which just is a very simple polysugar. And by generating the degradable layers underneath this siRNA layer, the idea was that you would get breakdown here underneath, and it would essentially slough off, and that's not a technical term, these thin films, and you get nanoplexes or flakes that will be delivered gradually to the cells that surround the bandage. So this is the coating of the scaffold or the nylon dressing. We're going to put down our degradable layers and then our siRNA layers. This is actually a focal microscope image of our layered wound dressing. It's nylon tegaderm, which is used by surgeons, and we coated it with siRNA. siRNA is labeled with a green fluorophore, and so you can see it very nicely. And if you go in and out of the page, or if this is actually zooming in and out towards you, you can see that it coats all of the different surfaces and crevices of the bandage. We can change the number of under layers and the number of top layers, and that yields differences in the total loading of the film, as you can see here, and also in the rates of release of the film. So we have a lot of tuning that we can do with these two sets of layers. We optimized those layers using in vitro tests and cell culture, and then we used a diabetic mouse model to test them. In this case, these are mice that intrinsically are diabetic. They're not induced. And for that reason, we can actually introduce these two wounds on the backs of the mice and look at the relative rates of wound closure. So we looked at both untreated wounds, wounds applied with the siRNA against MMP9, and wounds that contained siRNA that didn't encode for anything as a control. And what you can see on the right is the amount of gene expression of MMP9, where we normalize against the uncoded. We look at the control and we see little to no difference, but with the MMP9 dressing, we see that we have decreased MMP9 expression. That's MMP9 expression. If you actually measure the activity of the protein in the wound, here you can see MMP9 activity is also lower. This is after a 1-week period. After a 2-week period of the bandage on the wound, we see that we've lowered MMP9 expression down by about 85%. So we have a very real delivery of siRNA within the wound that is sustained over time. Because it's sustained over time, it allows us to see this gradual generation of collagen in the wound cavity. So in this case, collagen is seen as blue. And you can see the 7-day treated and untreated. And you can see the margins of the wound here and here. In the untreated, you don't really see this nice blue line of collagen deposition. But you do see that it's being formed here at the treated site. At 14 days, there's a more dramatic difference between the untreated and the untreated wound. And you can see a blow-up of this tissue here, very nice granulation tissue that is now ready to undergo that next step of remodeling. So we're excited about these results, and we're looking at how we might be able to further advance them. And we can also see rates of increase in wound closure in these animals. And some of the things that we have to do, if those who are curious, for translation of this, is to move it to a more realistic animal model. Rodents are resilient animals, and they always close their wounds, no matter what. So it's hard for us to go out in time the way you would with a real patient. However, pigs do wound quite a bit more like humans. So we're looking now at trying to get funding to look at this in pigs. In the meantime, we're also looking at a range of different targets, both for chronic wounds and also for another kind of wound that can go awry, and that is hypertrophic scarring. This is something that happens frequently with third-degree burns and with a range of very traumatic acute wounds, wounds that are in normal tissue. In these cases, the damage is so high that there is some change in the way that tissue is regenerated. And instead of having a case where you're trying to encourage the body to generate enough collagen, you have a case where you get stuck in that collagen-creating mode. And you generate too much collagen in the wound site, which can cause shrinkage at the wound margin, and of course these very evident scars. A very light and minimal version of this would be keloiding. But hypertrophic scarring on a large scale can lead to disfigurement and even some change in mobility for patients. So we're looking at ways in which we can use siRNA against those genetic targets. And these are going to require more sophisticated models. We've begun to work with Martin Yarmish at Shriners Hospital and with Jeff Gertner, who is at Stanford. All right, so saved the best for last. We're in the Koch Institute, which is focused on addressing cancer. And as a chemical engineer in that institute, I'm very interested in learning from my biology neighbors how we can better address cancer using our nanomaterials. One of the things that we've been particularly interested in are highly aggressive forms of cancer, the ones that tend to essentially return even after there's been a round of chemotherapy. The reason that this happens is because cancer in general, of course, is a disease of genetic mutation, and some of these genetic mutations can lead to inability to survive chemotherapy drugs, sometimes in very clever and frustrating ways. So when a patient is treated, for example, in ovarian cancer, there is a certain fraction of those patients treated who can end up with a new appearance of cancer cells that are resistant because they survived the first onslaught. We're interested in being able to design nanomedicines that can deliver a combination of drugs that eliminate the ability for resistance. One way of doing this is to take a nanoparticle that is going to be the carrier for our drug in the bloodstream. It's going to take it to the tumor. Inside that nanoparticle, we can incorporate some sort of chemotherapy drug, one that's known, one that we have come to rely on, but one that frequently undergoes or we experience resistance to. Around that nanoparticle, using layer by layer, we can put down a positively charged layer and then a negatively charged layer of siRNA or another nucleic acid that helps to lower the tumor defense mechanism, that genetic mutation that actually enables this resistance. We can then put down another positively charged layer, so we essentially have this siRNA sandwich, plus minus plus. And finally, we need another outer layer. The reason that we need this is because if you introduce a positively charged particle into the bloodstream, it will absorb a large number of the mostly negatively charged proteins in our blood. It will become recognized as a foreign object, and the body will very efficiently eliminate it within hours before it has any chance to get to the tumor. So we don't want that. We need some sort of stealth layer so that immune cells don't recognize this particle as something foreign. So this final layer is going to be negatively charged. The reason that helps is because most mammalian cells bear their own negative charge because of bound negatively charged glycans on their surface. So if you can think of other mammalian cells as being negatively charged objects in the bloodstream, and nanoparticle has a negative charge, they're going to be more repulsive than attractive, because like repels like. But beyond using charge just for that repulsion, there's an additional element that's important here. We have to choose a negatively charged polymer that has a large amount of water associated with it. If we can do that, then we essentially are introducing a nanoparticle that has a layer of bound water, and that bound water gives us stealth. We think of it as an invisibility cloak. It looks just like water. Don't look at me, you know, I'm just another water molecule. And the more we're able to do that, the less we lose the nanoparticle. Now, to be clear, in all nanomedicine, we will lose nanoparticles to excretion and clearance. So the name of the game is to push the balance more toward the tumor than to clearance. So in this modular design, there's one part of this nanoparticle strategy that's important to mention, and that's that the very size of the nanoparticle can help us get into the tumor. It turns out that when tumors are developing, they're developing very rapidly, and they're trying to generate vasculature to support the need of the tumor. However, in this rapid development of blood vessels, there are often small defects that are formed. And especially in early tumors, although also in some late tumors, it's thought that these defects are about the size of a few hundred nanometers. So if you have a nanoparticle that is 100, 120 nanometers or so in size, the belief is essentially that when you introduce the nanoparticle into the blood vessels, the nanoparticle will navigate the bloodstream, and if it can stay in the bloodstream long enough, will end up in the blood vessels that belong to the tumor. And when it gets into the tumor, it will go through those leaks and actually get caught in the tumor before it finds another leak to go out in. So when I talk to younger audiences, I say it's very much like a pinball machine. And then they look at me. Like, you know, and the ball goes up and it gets caught in the nooks and crannies, it rolls around, and finally it makes its way back down much later. And I think that they're still virtual pinball machines, but they sort of get. The general idea, though, is that you're increasing the residence time of the nanoparticle in the tumor by doing this. So here's a picture of mice. They have tumors on their backs, and you can see that at 24 hours, we used a quantum dot in this case and layered it with our layer by layer film, that our layered quantum dot particles are accumulated in the tumors. If you look at an earlier time shot, you would see that at first the entire animal lights up, and the nanoparticle is everywhere in the bloodstream. You can see that because it's in the bloodstream, it goes everywhere where blood vessels go, but it gets cleared everywhere but in the tumor where it hangs around. Now, once we get into the tumor compartment, we've used size to help us with that, and we've used stealth to allow us to circulate long enough to get in there. We need a way to get cells to actually take up the nanoparticle. And, you know, we spent all this time developing a stealth nanoparticle. So why would a cancer cell engage with this disguised nanoparticle? We have to either transform the nanoparticle when it gets into the tumor compartment, or label the nanoparticle with something very specific and precise that only tumor cells recognize. And we actually use both strategies in our lab. In this case, what we're showing is a nanoparticle on the right at pH 7.4, which is the bloodstream pH. And at that point, the nanoparticle is very strongly negatively charged, and it has this core, and hopefully you can see there's a little bit of a halo, a very thin halo, and that's our layer by layer film. This layer by layer film consists of poly-L-lysine. There's a polypeptide that has an amine group, and on top of that is hyaluronic acid, which is a native polysaccharide, glycosaminoglycan, that makes up part of our tissue matrix. But it has a large number of negatively charged carboxylate groups on it. So plus and then minus. It turns out that both of those two groups, for those who remember in light chemistry, you know, those two groups can undergo a change in their relative charge with pH. So what happens is that at pH 7.4, the combination of these systems blended into a layer is net negatively charged at pH 7.4. But as you begin to go to slightly lower pH, you're essentially erasing negative charge, because in chemistry talk, we are protonating the carboxylic acids. In regular talk, we are essentially eliminating that charge as we move down to lower pHs. And this becomes more of a neutral particle. So we lost some of that repulsion. Because we've erased the negative charge, and it's charge that holds the film together, the film kind of expands. It's trying to destabilize a little bit. So it gets fatter and softer. It turns out that cells like things that are neutral and are sort of soft like this. So we just naturally, because tumors are slightly lower in pH, just like wounds, we tend to get a nanoparticle that transforms when it gets into the tumor compartment and becomes much stickier to cells. So cells are now kind of interested in engaging this nanoparticle, and we see a big increase in how many of these nanoparticles are taken up by the cell when we go to the tumor compartment conditions. However, and this is actually just an image, again, I think the lighting is more difficult to see, but it's showing a tumor section, and the colored regions that you can just barely see are the overlap of nanoparticles with hypoxic regions of the tumor that are slightly lower in pH because they're not as good at getting oxygen to the tissue there. So we have size that helps us get in, and we have this tumor environment that sort of transforms the particle, makes it stickier, and helps the tumor cells to take up the nanoparticle. But as I mentioned, we like to have really more specific tags on our nanoparticle that tumor cells will take up and other cells won't. And it turns out that hyaluronic acid, the very same molecule that I mentioned before, binds a receptor that is present in very high densities on cancer cells called CD44. CD44 is a receptor that is on healthy cells, but it tends to be present on healthy cells during rapid periods of growth or development, or cell migration. However, it's not present in the same density as it is on cancer cells. And of course, you can imagine cancer cells are doing these things all the time, migrating, growing, proliferating, and they have large numbers of CD44 receptor on their surfaces. So it turns out that because we have this HA, hyaluronic acid layer, on the outside, we bind tumor cells very effectively. And I wish I could change the lighting, but I can't. But you can kind of see a little bit here that there's a little bit of glowing red. Is it possible for me to change the lighting? Thank you, Fred. Fred, by the way, is a postdoc who's been working in my lab in collaboration with Mike Yaffe, and is actually using these systems to address glioblastoma. Thank you. All right. So here you can see, hopefully a little bit better, that the nanoparticle is in cells here that overexpress CD44. We've labeled it red. So you can see the nuclei of the cells are blue. And you can see that you don't see the red in the cells underneath. And that's because we essentially titrated that with a large amount of free hyaluronic acid to compete out the nanoparticles. So we can show that this binding is very specific to the presence of hyaluronic acid. On the right, you see a tumor section. And in this section of tumor, again, blue is staining nuclei, and these are cells on the outside of the tumor. They call stromal cells, but the real tumor cells are inside. And we can stain for CD44, which is that receptor. And here it's shown in red, and you can see the nanoparticles in green, and that there's this orangey glow, and that's the overlap of the two. So we're essentially targeting these nanoparticles to tumor tissues using a combination of molecular tagging and changes in the tumor environment. Because we're using size, microenvironment, and the receptor, we call this sort of a triple targeting. All right, so we can generate this outer layer to get the nanoparticle where it needs to go. And now let's talk just very briefly about incorporating siRNA into this middle layer. We represent it as this blue color here. We looked at a bunch of positively charged polymers that could layer with the siRNA. And after optimizing, we found one that works really well, that is a simple polypeptide. Now that we have our nanoparticle constructed, we want to choose a target, and in this case we chose a target that we could understand easily. It was a gene that's been well characterized. It's the MRP1 protein. It is a pump, it's a protein pump. It actually sits at the cell membrane. And when one of these drugs, which are essentially designed as DNA damaging drugs, enters the cell, it rapidly gets pumped back out again. So it's actually a very clever mechanism. You can think of it as, again, like the feed the baby mechanism, in which you're trying to feed a baby something it doesn't like, and you spoon the food in and it comes out immediately. It's the same kind of idea here. So we're going to target the MRP1 protein. We want to silence it. And if we can silence it, then we should be able to make a tumor that normally would be more resistant to our DNA damage drug, less resistant. So we use doxorubicin, which is a very common DNA damaging drug. Doxil is a product that is essentially a form of doxorubicin. We place doxorubicin molecules inside a liposome. The liposome is negatively charged, so we have essentially this vesicle of chemotherapy drug. And then we put down those 4 layers on the surface of the nanoparticle. And of course, the middle layer is siRNA, and we want to make sure that we are knocking down the MRP1 protein. And on the left, we're using PCR to show that we go from the full expression of the MRP1 protein to something that is closer to a 70 to 80% reduction in the expression of the protein in the tumor. Here we're looking at sections and using a stain for the MRP1 protein, and we can see that it is present when we use a scrambled siRNA, but when we deliver siRNA against MRP1, we see that that color, that bleached look is gone. Again, we look at a very simple model of tumors on the backs of mice. We are going to use a triple negative breast cancer cell line to generate these tumors. And if we just treat with saline as a control, we can see that these tumors grow very rapidly. This is just a 2-week period. If we look at the nanoparticle that contains doxorubicin alone, we can see that we can slow the rate of growth of these tumors. But if you look at the relative rates of the control and the doxorubicin, we can see that they're still growing at a fairly rapid rate. They're about doubling over a period of 2 weeks. However, if we look at the combination system that has doxorubicin and siMRP1, we can actually see a regression in these tumors over the same 2-week time period. It's indicating that using this combination therapy, we can indeed control this 1-2 punch and gain some real progress against these really aggressive cancers. Since we're running short on time, I'll briefly explain this last example. It's essentially the same nanoparticle approach, but using a more realistic form of cancer, non-small cell lung cancer. It's based on a model that was developed here at the Koch Institute by Tyler Jacks, and it's representative of some common mutations that happen in more aggressive forms of non-small cell lung cancer, which include the presence of the KRAS oncogene, which is not druggable using normal inhibitor molecules. So we think the siRNA is a great way to turn off that gene. And also, it has changes in another gene called P53. P53 is normally a protective gene, but it mutates in this form of cancer so that you lose that protection. And we want to replace that function with a microRNA known as miR34. It's also a nucleic acid and is about the same size as siRNA. So in our package this time, we have a DNA damaging drug called cisplatin or platinum. And we have siRNA for KRAS and miR34A all absorbed in the blue layer. And everything else is the same as before. This drug releases so that siRNA comes out more rapidly, and cisplatin comes out more slowly once it's inside the cell. So again, this is going to be a more native kind of model. So the tumors are generated throughout the lung as many small tumors, and it's a model that can also become metastatic. Here you can see tumor targeting. These are healthy mice, and these are mice with lung cancer. You can see this bright light up here in the lungs of the mice. If you actually excise the organs, you can see that the lungs of the tumored mice are really lit up with our nanoparticle compared to the lungs of the healthy mice. And you can see this quantitatively in the bar graph. And finally, again, this is a very aggressive model. We can use a measurement of the tumor mass in the lungs and also see that there's a decrease in the size of the tumors when we have the combination therapy compared with just the cisplatin or just the RNA. And finally, we can look at the survivability of the mice, and we can see that we have enhanced survivability or lifespan of these highly tumored mice by about 30%. So we're excited about the fact that we can actually see a meaningful impact in what is a very aggressive model. So I will stop here. I'll say we are working on other forms of cancer, like lymphoma. And we're also working with my good friend Angie Belcher, also here at the Koch Institute, to combine our work with near-infrared imaging so that we can not only treat these tumors, but see them at very early stages. So I'll skip ahead and just conclude by saying that this is a very versatile technique, and it allows us to address everything from bone regeneration on very large length scales, to soft tissue wound healing, to addressing cancer using the very smallest scales, nanometer scale systems, to deliver therapeutics in a very controlled fashion. And this essentially is a very modular approach that can be applied to a range of other applications as well. Today I talked about the work of Eric Drayden, Jason Deng, Lee Gu, Nisar Juha Stephen, and this is a list of other group members in the lab and my collaborators, including Martin Yarmish and my inspector for the soft and the hard tissue. Angie and Michael are collaborators here at the Koch on cancer, as well as a range of other collaborators. And I'd like to thank you for your attention and your patience, and welcome any questions that you may have. Thank you.
Thank you. And the way you explained it, I almost understood it. Can we have some questions? I think one in the back. Okay. Sure.
I have two questions. They're pretty closely related. Number one, how long does it take from identifying a promising technique to actually getting it implemented, and say licensed by the FDA? And the second question, which is almost the same, are any of them close to that qualification at this point? Thanks.
Absolutely. There are a range of answers to the first question. I've seen technologies developed here at MIT and elsewhere that have been developed over a short, what we consider a short time period of 3 or 4 years. And other technologies, I know one example in which I was the thesis committee member on a concept introduced in my first year as a faculty member by one of my colleagues, Bob Langer, and I saw, and Michael Sema, and I saw that become a product just a couple of years ago. So it can be 20 or 25 years if there are, different forays that happen. But I would say on average that it's somewhere between 7 and 10 years if you're talking about a complex biomedical approach. And the reason for that is that at least there's time to develop the approach, and you need time to be able to demonstrate it in well-tested models in animals. Then there's a period when you're trying to translate it. And during that period, there's typically some fundraising that has to go on. To do trials, you usually need a partner, both either a strong clinical partner or a company. And so all of these processes typically take time. So I think 7 years might be a more fair answer to that. In the work that we're doing, we actually have a company called Layer Bio that is working on the larger scale problems. Right now they have funding and a partnership with Novartis, and they also have some newly burgeoning partnerships with other companies. And they're looking at some of the different forms of wound healing or tissue regeneration in this work. So that is an example that company was founded in 2013 and was the result of interactions with a collaborator that I met maybe 3 or 4 years before that.
Thank you. Any other question?
Thank you very much for a fascinating talk. Going back to the LBL assembly, in my mind I was equating it to how semiconductor chips are created. So would you comment on what technology used to create an LBL assembly?
Absolutely. Process is also part of it. So in our work, we first began generating these layer by layer films by dipping, and you saw the picture with the beakers, and we were using robotic slide stairs. We still do some of that work. However, it turns out that you can generate the layers much more rapidly using spray layer by layer assembly. Convection decreases the time period needed for exposure to solution from several minutes to a few seconds. I did have a student who helped to develop an automated spray layer by layer approach, and he actually did start a company that was not a biomedical company. That time frame was a little different. It was over 3 or 4 years, and then he sold that technology to 3M, but still holds some patents on it. That makes the process more rapid, but if you're going to work with a precious component like siRNA, it needs adaptation, and I've had some of my students who have looked at how to preserve components while you're generating these thin films by recycling. For the nanoparticles, we actually went from adsorption and centrifugation of colloids to now what is a tangential flow filtration membrane approach for nanoparticle isolation steps. And that also has greatly sped up the process.
Wonderful talk. A couple of questions. First, from the first part of your talk, what you showed was an application to surfaces like an implant or a surface wound. How would you adapt this technology, say, for a deep cavitary type wound or organ damage?
That's a great question. One of the things we've been thinking about is actually coating microspheres that are degradable with our layer-by-layer films. It's something that we've done for other projects, but for wound healing it actually would be a great fit because you can actually generate beads that can be poured into the wound and would be conformal. And this would work especially well for deep puncture.
And thank you. And then the other questions dealt with the last part of the talk. You had shown in one of your examples using paclitaxels, your drug, and I think to a number of people in the audience it wasn't lost that might be taking head-on albumin nanoparticle paclitaxel or Abraxane. What would be the advantages of this technology over that?
Oh, thanks for asking. Abraxane is a colloidal suspension that uses albumin secondary interactions, hydrophobic interactions with paclitaxel, which is a hydrophobic molecule. It's on the verge of perhaps being called a nanomedicine, but isn't quite. It's a colloidal suspension injected in the bloodstream. Its half-life is greatly shortened by the fact that it is not a very highly stabilized colloid. So rather than getting, I would say the residence times that we get in our nanoparticles are somewhere between 18 to 24 to 28 hours half-life, Abraxane has a much shorter few-hour half-life. For that reason, you have to introduce it in larger quantities. The other difficulty with Abraxane is that it does have, it is an improvement over the original formulation, but it still has fairly large exposure in terms of toxicity. So because it's not a very highly stable colloid, the drug is able to interact with cells throughout the body. I didn't have time to put it into this talk, but one of the papers that we did with Mike Yaffe was essentially looking at a very simple combination of 2 inhibitors, which together, injected into the bloodstream, are quite toxic. You can see a large amount of liver toxicity, for example. But in these vesicles, essentially, you protect the rest of the body to a large extent from these side effects, and therefore you get a much lower side effect and a much lower toxicity. That allows us to dose patients with combinations that weren't touchable before. But for a drug like paclitaxel, it means that we can use less drug for the same effect.
Any other question?
Just wondering if this layer-by-layer technique could also be useful outside of the body on scaffolds for growing tissue and organs.
Yeah. So that is something that is interesting to us. We have looked at layer-by-layer systems as a potential place for regenerative systems or maybe organoids. And we have some collaborations, although we haven't introduced the release of growth factors into those collaborations with people like Linda Griffith, who are interested in building 3D tissue mimics. I think the next step beyond would be to have a fully functioning organ. I think that's very interesting. Although we don't have any direct work in that area right now, we have friends and colleagues who work in that area. We're in constant communication about that.
Nothing for the hair or something like that?
Now, that would be my hope.
Thank you very much. Maybe, Megan, you want to close the session?
I just want to say thank you again for a fascinating lecture, and I'm sure everybody has still got more questions, so we're hoping people to stick around and join us for some cookies and more drinks outside. And I just want to present you with our a small token of our appreciation, insulated lunch sack and a wine glass that has been carefully wrapped layer by layer.
Thank you.
Thank you. Thank you.